Hi, and welcome to the Smell Podcast. I'm your host, Katie Price. Today I wanted to share with you all the story of two very important scientists. Their names are Richard Axel and Linda Buck. So we'll start off by learning more about each of these scientists, and then we'll talk about their contribution to science, specifically how they contributed to the understanding of the science of smell. All of the following information was taken directly from Wikipedia, so shout out to them, and I'll make sure to include the URL to the site so you can see my sources and find that information later on if you're interested in learning more. Linda Buck was born in Seattle, Washington on January 29, 1947. Her father was an electrical engineer who spent his time inventing and building different items, while her mother was a homemaker who spent a majority of her free time solving word puzzles. Buck was the second of 3 children, all of them being girls. Buck's parents raised them to believe that they had the ability to do anything they wanted with their lives, and she attributes her affinity of science to her parents' interest and the dedication she grew up around. In 1994, Buck met Roger Brent, who's also a biologist, and the two married in 2006. Buck received her Bachelor of Science in Psychology and Microbiology in 1975 from the University of Washington, Seattle, and her PhD in Immunology in 1980 under the direction of Professor Ellen Vitetta at the University of Texas Southwestern Medical Center at Dallas. Side note, microbiology is a branch of biology dealing with microscopic forms of life, and immunology is a science that deals with the ways in which the body protects itself from diseases and infections. In 1980, Buck began postdoctoral research at Columbia University under Dr. Benvenuto Pernis. In 1982, she joined the laboratory of Dr. Richard Axel, also at Columbia in the Institute of Cancer Research. After reading Sol Snyder's group research paper at Johns Hopkins University, Linda Buck set out to map the olfactory process at the molecular level, tracing the travel of odors through the cells of the nose to the brain. Side note, olfactory means of, relating to, or connected with the sense of smell. Buck and Axel worked with rat genes in their research and identified a family of genes that code for more than 1,000 odor receptors and published these findings in 1991. Later that year, Buck became an assistant professor in the neurobiology department at Harvard Medical School, where she established her own lab. After finding how odors are detected by the nose, Buck published her findings in 1993 on how the inputs from different odor receptors are organized in the nose. Essentially, her primary research interest is on how pheromones and odors are detected in the nose and interpreted in the brain. She is a full member of the Basic Sciences Division at Fred Hutchinson Cancer Research Center and an affiliate professor of physiology and biophysics at the University of Washington, Seattle. Okay, next up we have Richard Axel. Again, this information was taken straight from Wikipedia. Axel is married to fellow scientist and olfaction pioneer Cornelia Barkman. Previously, he had been married to Anne Axel, who is a social worker at Columbia University Medical Center. Owing to his tall stature, Axel played basketball during high school. This is a fun fact. Axel once played a basketball game in high school against Lou Alcindor, who later became known as Kareem Abdul-Jabbar, who went on to become one of the greatest basketball legends ever. Axel scored 2 points in that game, and Kareem Abdul-Jabbar scored 54. Born in New York City, New York, Axel graduated from Stuyvesant High School in 1963. He received his A.B. in 1967 from Columbia University, and from what I understand, an A.B. is like a Bachelor of Arts. He also received his M.D. in 1971 from Johns Hopkins University. He returned to Columbia later that year and became a full professor in 1978. During the late 1970s, Axel, along with microbiologist Saul J. Silverstein and geneticist Michael H. Wigler, discovered a technique of co-transformation via transfection, a process which allows foreign DNA to be inserted into a host cell to produce certain proteins. A family of patents, now colloquially referred to as the Axel patents, covering this technique were filed for February 1980 and were issued in August 1983. As a fundamental process in recombinant DNA research as performed at pharmaceutical and biotech companies, this patent proved quite lucrative for Columbia University, earning it almost $100 million a year at one time. and a top spot on the list of top universities by licensing revenue. The Axel patents expired in August of 2000. Axel's primary research interest is on how the brain interprets the sense of smell, specifically mapping the parts of the brain that are sensitive to specific olfactory receptors. He holds the titles of University Professor at Columbia University, Professor of Biochemistry and Molecular Biophysics and of Pathology at Columbia University's College of Physicians and Surgeons, and Investigator of the Howard Hughes Medical Institute. In addition to contributions to neurobiology, Axel has also made seminal discoveries in immunology, and his lab was one of the first to identify the link between HIV infection and immunoreceptor CD4. In addition to making contributions as a scientist, Axel has also mentored many leading scientists in the field of neurobiology. 7 of his trainees have become members of the National Academy of Sciences, and currently 6 of his trainees are affiliated with the Howard Hughes Medical Institute's Investigator and Early Scientist Award programs. Alright, now that we know more about Linda and Richard, let's get to the reason I'm talking about them today. They were both jointly awarded the Nobel Prize in Physiology or Medicine in 2004 for their research on how the olfactory system works. Remember, olfaction means something to do with smell. So I'm going to read the following article to you all. This article is called Richard Axel and Linda Buck awarded 2004 Nobel Prize in Physiology or Medicine, and it was published on October 4th, 2004. I'll include the link to the article in the episode description so you can read it later if you want. In 1991, Axel and Buck, who was then a postdoctoral fellow in Axel's lab, discovered a family of genes that encode the odorant receptors of the olfactory epithelium, a patch of cells on the wall of the nasal cavity. The olfactory epithelium contains some 5 million olfactory neurons that send messages directly to the olfactory bulb of the brain. When an odor excites a neuron, the signal travels along the nerve cell's axon and is transferred to the neurons in the olfactory bulb. This structure, located in the very front of the brain, is the clearinghouse for the sense of smell. From the olfactory bulb, odor signals are relayed to both the brain's higher cortex, which handles conscious thought processes, and to the limbic system, which generates emotional feelings. Each olfactory neuron in the epithelium is topped by at least 10 hair-like cilia that protrude into a thin bath of mucus at the cell surface. Somewhere on these cilia, scientists were convinced there must be receptor proteins that recognize and bind odorant molecules, thereby stimulating the cell to send signals to the brain. The receptor proteins would be the key to answering 2 basic questions about olfaction, explained Axel. First, how does the system respond to the thousands of molecules of different shapes and sizes that we call odorants? Does it use a restricted number of promiscuous receptors or a large number of relatively specific receptors? And second, how does the brain make use of these responses to discriminate between odors? The string of discoveries that totally changed the study of olfaction resulted from a new emphasis on genetics. Instead of hunting for the receptor proteins directly, Axel and Buck looked for genes that contained instructions for proteins found only in the olfactory epithelium. Their efforts produced nothing at first. Now we know why our initial schemes failed, said Axel. It's because there are a large number of odorant receptors, and each was expressed only at a very low level. Finally, Buck came up with what Axel calls an extremely clever twist. She made 3 assumptions that drastically narrowed the field, allowing her to zero in on a group of genes that appear to code for the odorant receptor proteins. Her first assumption, based on bits of evidence from various labs, was that the odorant receptors look a lot like rhodopsin, the receptor protein in rod cells of the eye. Rhodopsin and at least 40 other receptor proteins crisscross the cell surface 7 times, which gives them a characteristic snake-like shape. They also function in similar ways, by interacting with G proteins to transmit signals to the cell's interior. Since many receptors of this type share certain DNA sequences, Buck designed probes that would recognize these sequences in a pool of rat DNA. Next, she assumed that the odorant receptors are members of a large family of related proteins, so she looked for groups of genes that had certain similarities. Third, the genes had to be expressed only in a rat's olfactory epithelium. Had we employed only one of these criteria, we would have had to sort through thousands more genes, said Axel. This saved several years of drudgery. Buck recalls that I had tried so many things and had been working hard for so many years, with nothing to show for it. So when I finally found the genes in 1991, I couldn't believe it. None of them had ever been seen before. They were all different, but all related to each other. That was very satisfying. The discovery made it possible to study the sense of smell with the techniques of modern molecular and cell biology, and to explore how the brain discriminates among odors. It also allowed researchers to pull out the genes for similar receptor proteins in other species by searching through libraries of DNA from these species. Odorant receptors of humans, mice, catfish, dogs, and salamanders have been identified in this way. The team's most surprising finding was that there are so many olfactory receptors. The 100 different genes the researchers identified first were just the tip of the iceberg. It now appears that there are between 500 and 1,000 separate receptor proteins on rat and mouse, and probably human olfactory neurons. Wow, that was a lot of sciencey stuff. Thank you to the Howard Hughes Medical Institute for their permission to read this article. So basically what I'm understanding is that odors are molecules, and they go up in our brain and are received there. We know this now because of the work of Richard Axel and Linda Buck. Once they're received by us, that signals our brain that we're smelling chocolate, mint, cut grass, etc. I told you all before that I would share my personal story on another podcast, and I still plan to do so, but for now I wanted to share this about myself. The reason I have anosmia is because my olfactory cells don't receive the odor molecules. Someone once told me, and I can't remember exactly who, but I think it was probably one of the many doctors I went to see, that you can think of smelling like a lock and a key. The lock is the olfactory cells in your brain, and the keys are the odor molecules. I've got broken locks, so the keys don't fit. So, what do you guys all think? Did I get all of that way wrong? Am I right on track? Please send me your thoughts to thesmellpodcast@gmail.com, or you can tweet me at thesmellpodcast. You can find the sources and links to articles for today's show in the episode description. Please rate, review, and subscribe to The Smell Podcast on Apple Podcast or iTunes so others can find the show. You can listen on iTunes, Stitcher, Spotify, and many other places. So until next time, thanks for listening and have a great day.